start?
I think they're working.
Yeah. All right. Good morning, everyone, and welcome. This is obviously one of the most exciting sessions of this EHDN meetings, which is in its entirety dedicated to an update on the status of the ongoing clinical trials. So my name is Ahmad Aziz. I am a neurologist from Bonn, and until yesterday I was the chair of the Scientific and Advisory Committee of the EHDN, and together with my esteemed colleague Tiago Mestre, who is an associate professor at the Ottawa University in Canada, we have the privilege to chair this session. As you've seen from the program, we have tried not to be too restrictive with our selection of clinical trials, to give as many companies as possible to share their exciting results with us. However, as a consequence, we have a very tight schedule. So I urge all the speakers to abide by the time. So they have 7 minutes for the presentations, followed by 3 minutes of discussion and Q&A. And with that, I hand over to my colleague.
Thank you. And so, good morning, everyone. As Ahmed said, it's a pleasure and I think an honor that we're able to host such a nice, I'll call it a menu of clinical trials. And so without further ado, I open the session with Dr. Raul Insta, founder and CSO of the SOM Biotech. Thank you.
Thank you very much. Thank you very much, Tiago. Good morning, everybody, and thank you for coming so early in the morning today. It's going to be a promising morning. So I'm going to talk about SOM 3355, our product for Huntington disease, which was discovered and is under development by the company SOM Biotech, a small company based in Barcelona in Spain. We have an artificial intelligence platform, which is called SOM AI Pro. And through this platform, we can identify new activities of known drugs. So we repurpose drugs into new indications, mainly in the orphan space. So by doing this, we took tetrabenazine some years ago in order to improve the safety of this drug. And we discovered another one called bevantolol, which is SOM 3355. Bevantolol is a beta blocker that has been developed and used in the United States, Europe, and Japan. Right now, it's only available in some Asian countries for the treatment of hypertension. But this drug has an atypical VMAT2 inhibitor profile that you can also see in a poster that we have, which is G1414, okay, because it binds in a different way to the VMAT protein. So what we did is we validated in vitro and in vivo the activity of the drug, and we planned directly for a phase 2A in 32 patients in order to see if it's working on this disease. We could skip many preclinical and phase 1 development period because the drug, as I said, is already known. And right now we have just completed a phase 2B, which I will explain later. So these are the results very quickly of our phase 2A. In our phase 2A, as I said, there are 32 patients. Okay, it was done in Spain in 4 hospitals around the Barcelona area. And we got around 60% of the patients had improved more than 2 points in the total maximum chorea score, which is the primary endpoint. Usually it's the primary endpoint in chorea and Huntington disease, in relation to placebo, in comparison to placebo. And around 78% of the patients improved more than 2 points from baseline. And it was statistically significant with the higher dosage that we used, which was 200 milligrams BID. So that's why we took the decision to jump directly into phase 2B. The safety was very good. This drug has been given for more than 25 years to healthy and hypertensive population, and more than 2.5 million patients have taken it. So the phase 2B that has just finalized, I have to say that it has been a success, okay, and that right now we are analyzing the data. So today I'm not going to present much data, but I will tell you about the next step. This study is a phase 2B double-blind placebo-controlled study with 3 arms, placebo, 200 milligrams BID, and 300 milligrams BID, with a treatment period of 12 weeks. Okay, 3 weeks to scale up the dosage, 7 weeks of treatment, and 2 weeks to go down. 23 sites in Europe have participated, 7 European countries, and we have recruited 171 patients. 140 of them have been randomized to any of the 3 arms. Okay, last patient, last visit was done in June, and the database lock was done by the end of July. So from August, we have been receiving data, and we are analyzing that data right now. So it has been a long, I would say, period, but successfully we have completed the trial thanks to our investigators and, of course, to the associations implied. Just a little bit remind about inclusion and exclusion criteria, which are quite standard for this type of studies. Okay, patients more than 21 years old, diagnosed of Huntington disease, okay, that have a total maximal chorea score of more than or equal to 10, plus a functional score of more than 7, or at least 7. Okay. Exclusion criteria mainly was the juvenile form of the disease, and also the controversial taking of neuroleptics and other medication. We allowed at the end the investigators to include some patients with neuroleptics, but fortunately very few of them, because as you know, this kind of drugs can interfere with the chorea treatments of another drug. So the primary objective, as I said, was to have activity versus placebo, okay, in the total maximum chorea score, okay, in any of the 2 dosages, 200 and 300 milligrams. Second objectives were safety, to confirm the safety of the drug, okay, tolerability and efficacy on other scales, clinical global impression of patient global impression of improvement and so on. Percentage of responders, percentage of improvement of the total maximal chorea score, and so on. Also, we have done, and we are receiving the data on that, PK studies on the blood samples of the drug, and also on prolactin, which is a biomarker of the mid-2 activity of the drug. We have predefined the analysis that we are doing right now on the protocol. It's established on the statistical analysis plan. Okay, and this predefined analysis includes, of course, the demographic data that I will show to you right now. Okay, and the DMC score changes from baseline in relation to placebo. It's through a mixed model, also ANCOVA. Okay, and the DMC analysis on different baseline levels of severity of the disease. Of course, safety, suicide scales, depression scales are also included because, as you know, this is the relevant point in this type of drugs. And the structure has to be validated. So what can I say today? Today, unfortunately for you, I can also only talk about the demographic data of the trials, okay? We're very happy with the results in safety and efficacy, but we're not prepared yet to show them to the community. So as I said, 171 patients have been screened, 140 randomized. Okay, it has been a randomization quite equal per the 3 groups in terms of gender. Almost half of the population are males and half females. The age is around 57 years old. Okay, and you can see here what's the total maximum chorea, for instance, mean, which was 10.5 at baseline. Sorry, total maximum chorea was 12.8 at baseline in general. Okay, total motor score was 36, and total functional scale was around 10.5. So right now we are analyzing this data. We plan to make an investigators meeting with all the sites next month, as soon as we close all the PK data, which is the last one, pack of information we want to receive. And after this investigator meeting, we will publish the results, and we'll make details more available in the next Huntington Study Group Congress in Cincinnati in November. What I want to confirm here today, and this is very important for us, is that we still want to strengthen our commitment to the Huntington disease community, patients and family. So we want to thank you very much, all the associations, of course, the Huntington Disease Network, the Enroll Group, and all the sites that have been participated in this clinical trial during these last 3 years. Thank you very much.
We'll have time for one question from the audience.
Yeah, so maybe I can ask a question. So thanks for your presentation. I was wondering, so as you know, there are 3 other VMAT2 inhibitors on the market, so tetrabenazine, valbenazine, and deutetrabenazine. So what's the main advantage of your compound in comparison to those others?
Yes, the VMAT2 inhibitors that are in the market, as you know, there's only one approved in Europe, which is tetrabenazine, and 3 in the United States, tetrabenazine, deutetrabenazine, and valbenazine. All the drugs are related to tetrabenazine, okay? And our drug, bevantolol, chemically structure is not related to tetrabenazine. That's why this drug binds to another site of the VMAT2 inhibitor, also has an activity on binding to a VMAT1, and the safety profile is totally different. The other drugs do have a black box in the leaflet because of suicide, depression, neuroleptic syndrome, and Parkinsonism, and so on. And we do not expect to have this black box in our leaflet because of the safety of the drug. So we expect to have an equal efficacy drug without less safety concerns.
Okay, thank you very much. Okay, then we can move on to the next presenter, who is Dr. Michael Hayden. He's Chief Executive Officer of Prilenia. So welcome, Michael.
Just come back one. So good morning. Really thrilled to be here, have this opportunity to reconnect with so many friends and colleagues, and to give you an update on where we are with information with regard to the development of predopidine. So today I'm going to share just 2 quick updates, not too much time. The first is we now have some open label data that we can continue to present, 2 years persistence of efficacy. And then secondly, I'm going to show you some data on the positive effects of predopidine not being masked by lower doses of certain antidopaminergics. And we continue to learn about this drug. Of course, to remind you that we've already shown before that predopidine can have, on the right-hand side, what you can see is improvement from baseline in this particular group with continued benefit over placebo all the way out to week 78. Today, I'm going to show you data that goes beyond week 78 in terms of the open label. So the open label initial analysis was really to look at the persistence of beneficial effects. The primary efficacy was off ADMs, subjects who completed 78 weeks. I'm going to show you just the persistence effect in various independent measures, and this is compared to propensity-matched historical control. This was TRACK-HD, which also had longitudinal data on Q-motor. So here we have the data on cUHDRS. What you can see is that pretty stable all the way out to 2 years. So this was encouraging for cUHDRS, a decline of minus 0.54 at 2 years, and as everybody knows, that would be slower than expected. And so how does it compare to a matched historical control? We had, through the kindness of Sarah Tabrizi and also Doug Langben, we had the database for TRACK-HD and looked at people who were propensity matched around many measures, and there were a total after propensity match of around 58 patients who had 2 years longitudinal data. What you can see are compared cUHDRS to this particular historical control is certainly significant difference compared to TRACK-HD for predopidine now over 2 years. And I'll just quickly show you some of the other measures as well. For example, this is TFC. Again, no real significant decline, persistence of effect compared to TRACK-HD. Again, benefits relative to that historical control. If you look, and this was important because we wanted to look at Q-motor, and this was finger tapping OII, but this was seen throughout all measures of Q-motor, and here you can see significance. So the summary here is that looking at this data, one sees that there is persistence of effect in terms of slowing of progression, as would be expected in the natural history of Huntington disease. This is Stroop, for example. Again, no real deterioration at 78 weeks compared to TRACK-HD, where we see this kind of deterioration. So this was encouraging. We see persistence of predopadine's effects up to 2 years. Let me show you also some, I'd say, surprising results on the positive effects of predopidine not being masked by lower doses of certain ADMs. A few basic facts. Predopidine is a CYP2D6 inhibitor. And here's data, for example, using metoprolol that you can see here, where if you give metoprolol, which is a CYP2D6 substrate, after exposure with predopidine, you get a very significant increase in exposure. So the issue is many ADMs are metabolized by CYP2D6. What would we expect with some of these? So the regulatory labels actually provide specific guidance for dose reductions when given with CYP2D6 inhibitors such as predopidine. Let me just show you. Aripiprazole. They recommend going half, this is from the label, FDA approved. Trial dose normally for adults around 10, half the dose would be 5. We defined lower dose based on the regulatory labels, 5 milligrams. Same with deutetrabenazine, should not exceed a single dose. We defined this as 30 milligrams. And then the question was, if you use lower doses of some of these, how does this compare to patients off ADMs? Just to note, we also did PK studies. Here you can see the metabolism, for example, of Austedo. I know the drug. I took it to approval. And what you can see is predopidine essentially would inhibit the further metabolism from active to inactive products. In the presence of predopidine, you'd expect more activity. Here you can see, this is just showing the scatter plot, that predopidine compared to placebo in the same study increases the plasma levels by 1.5 to 2-fold. And so the question is, do they maintain the treatment benefit? If you look at those patients who are on lower doses, and there was a small proportion, about 30% of patients who took ADMs were on lower doses as defined by the labels. Just to remind you, we looked at the mean difference, LS mean difference. It has been shown in the beautiful paper by Schobel et al. That a clinical decline of about 0.2 or a slowing of 0.2 to 0.3 years can define clinically efficacious treatment. Here you see predopidine essentially off ADMs, and here we see low dose. Low dose really looks very similar to predopidine off. Comparing this to high dose, quite different. So this is really giving us encouragement that predopidine can be used on lower doses of certain ADMs without masking the effects. So the summary is that predopidine shows long-term sustained efficacy up to 2 years with multiple independent measures. And this is confirmed to the matched cohort. ADMs can be used with predopidine, and the positive effects of predopidine are not masked by lower doses of certain ADMs. And let me just say, we are continuing to learn new aspects of this drug, and we will continue to share these findings and insights with the community to advance the care of patients who urgently need and deserve better treatments. Today, we're at a point where through this community's participation, focus, commitment, we now have a regulatory filing under review for a drug that, as you can see, has the potential to slow the course of the disease. This reflects a testament for so many people here to a long commitment that have embraced risk, made some thoughtful decisions, and stayed relentlessly focused. And so just to share with you, this has impact not just for predopidine, but also for patients on any other study. And this is a poster I'd go to. This is from Enroll. Jeff Long and others have looked at this. And I just want to say thank you. Thank you to the whole community. Thank you to the PIs, Ralph, Andy, Anne, and Sandra, to Sarah and Doug for access to the TRACK-HD, to EHDN for their endorsement and support, and of course the HD patient advocacy groups and all my colleagues at Prilenia. Thank you.
Well, thank you very much, Michael, for this update. Are there any questions? We have time for one or two questions. I think it was, yeah.
So we have 2 minutes.
Yeah.
And if she has discussed her personal view of your results with the research team.
We have read. We were quite surprised. Nobody ever discussed this with us. We come from a place of supporting all drugs in development for HD. We believe that patients with Huntington disease deserve multiple options. We will choose a suitable, appropriate way to respond. I won't really comment further, but just to say, obviously, we disagree. You know, when something gets to the regulators, this doesn't happen by chance. This happens because the regulators have done a pre-assessment, they invite a submission, it goes through review, and then eventually is accepted for review. It's important to note, it's not accepted for approval, it's accepted for review that can take a year. We support all efforts from families and patients and drug companies to find multiple options that in the end might be used in combination in an effort to have some impact on the suffering of patients and families with Huntington disease around the world. This has been a commitment I personally made for nearly 45 years, and that commitment remains steadfast, focused, and relentless.
Okay, thank you. So one last question.
Thank you, Michael. It's really encouraging to see the ADM data, and of course another very common class of drugs that these patients are on are the antidepressants. So do you have any sight of how pridopidine performs?
Yes, thank you, Anne. Really, I'm saying with some relief that there is no negative interaction between pridopidine and all the antidepressants. This is work that Jeff Long and others have done. We've also looked in PROOF, but also in Enroll, and antidepressants do not appear to have some of the potential adverse events. This is not to say that patients shouldn't be treated with ADMs. We have never said that, but certainly there needs to be care around that, as there would be with any drug. And what we are saying and seeing, that antidepressants do not have any effect on some of the clinical measures that would be to define progression and various other measures.
Okay, so thank you very much for the nice clarification. So we have to move on to the next speaker.
So switch to a different compound, a different mechanism of action.
Okay, then we move on to the next speaker, and it's a pleasure to welcome Dr. Ed Wild from University College London, who will update us on the uniQure trial. So welcome, Ed.
Let's do this. Bonjour encore, me again. Serious scientist mode. I'm too... They turned off this... Oh, I don't know. Here we are. Okay. I'm too short to be behind the podium. But I'm delighted to be here presenting an update on behalf of uniQure on the AMT-130 gene therapy program. I'll just give you a few seconds to copy this down into your notes. This is the standard pharma disclaimer about forward-looking statements. These are my disclosures. I'm an advisor to uniQure and a PI at the UCL site in this trial. I'm an advisor to numerous companies trying to develop treatments for Huntington's disease, including 4 of the other people and companies you'll be hearing from during this session. So AMT-130, as you hopefully know, is the first gene therapy for Huntington's disease. It is a surgically administered, stereotactically administered AAV virus containing a construct that encodes a Huntington-lowering RNA, which should reduce the production of Huntington protein. Importantly, the target sequence for the RNA is within exon 1 of the Huntington transcript. And so, in principle at least, it should lower all forms of Huntington protein, including full-length and the particularly toxic exon 1 fragment or splice variant. The program was launched in 2019, so it's, as one would expect for a novel gene therapy, a slow-burning program, but is gradually accruing a wealth of data. In the US, 26 patients have been enrolled, and that arm of the study has a sham control which runs to 12 months, after which patients are offered the opportunity, if eligible, to roll over onto active treatment. In the UK and the EU, which enrolled later, the sham was not permitted, and so that study is an open label. But in both territories, low and high doses of the gene therapy construct are being tested. And I'll be presenting the latest data cut, which is 2-year data from 21 patients. So if the sham only runs to 12 months, there's no possibility of a direct comparison with the 24-month data. So what we have the options for are fairly limited, because Enroll-HD, for instance, which is obviously fantastic and huge, doesn't have MRI scanning and therefore cannot be used for the striatal volume inclusion criteria for selection of a matched natural history cohort. It would be difficult, I think, for the sponsor to enroll such a cohort with long follow-up but no expectation of treatment. So instead, what's been performed is a propensity-weighted historical control cohort, very similar to what Michael described, which Prilenia have done. And this is a method that has widespread favorable recognition by regulators. It permits long cohorts to be well matched, both sort of physically and statistically. But of course, it doesn't account for potential placebo effects. But with very long-term treatment, as in this program, one would hope that those would extinguish over time. So in principle, and I'll be very quick here, you have some patients in your trial, you have a larger number of patients from a real-world cohort, and some of them are well-matched and some of them are less well-matched. And you choose the ones that match best, but then from those, the method upweights, it increases the effect that the well-matched participants have, and it appropriately diminishes the impact that the less well-matched participants have. So you end up with a cohort that includes as many people as possible. No one's excluded if they meet the inclusion criteria, but the degree to which they match is used to weight the data. And data from TRACK-HD and PREDICT-HD were used. The striatal volumes were used as to gate entry into the cohort, and 154 patients were selected. They already had well-matched baseline characteristics, but the propensity weighting increases the validity of the comparison. So on to the results at 2 years, relatively small numbers, and all p-values are nominal. So in the external controls, about a 1-point decline in cUHDRS over 2 years. In the low dose, that was 0.7. In the high dose, it was 0.2. And that was nominally statistically significant. Here's the time course data, and you can see the way that the gray, which is the natural history, declines over time, and then the purple is low dose, the blue high dose, stacking up in the order that would be expected for a dose-response relationship. Don't really have time to go into the breakdown, but here they are. And these data are also visible on the uniQure website. One interesting thing to contemplate here is the SDMT in the bottom right, where that's the one with the biggest difference between low and high dose. And in the preclinical data using animals with large brains, an important observation potentially was that striatal administration produced significant cortical transfer of the construct. So the other finding, which is particularly exciting to me, is that, as we all know, if you put instruments into someone's brain, you get a significant release of NFL, and that can take a long time to settle down. In the 12-month data, the low dose was already at baseline. In the 24-month data, both of the point estimates are below baseline. And to put that into context, my HD-CSF study run at UCL showed that in Manifest HD, you expect a 10 to 15% increase on average per year. And overall in this study, 76% of patients had an increase over 2 years. So it's really unusual for a group of patients to end up with a lower NFL 2 years later than it was at baseline. But this is what we've seen in this study. Patients from the HD-CSF natural history study who match the patients in the AMT-130 trial experienced a 26% increase over 2 years. AMT-130 pooled dose patients had a decrease of 11%, which was nominally different, significantly different from the baseline value. So that's a result I don't think we've seen before. Safety is looking good, particularly considering the mode of delivery. Some SAEs in the high-dose group, those were largely neuroinflammatory and confined to the weeks and months following administration, generally manageable with the usual treatments for neuroinflammation. And the new cohort, which is now enrolling, will have an immunosuppression protocol to hopefully suppress that inflammation, reduce the NFL peak and shift it to the left so that hopefully benefits can start to emerge earlier. So in summary, favorable trends on clinical measures, suggesting a reduction in the rate of decline, favorable NFL results, good-looking safety with manageable liabilities, and RMAT designation by the FDA, which is essentially a fast-track designation for advanced therapeutics like this. It's actually the first the FDA has given. This enables more frequent conversations around things like future plans and potential approval pathways. Cohort 3, which is the new cohort with immunosuppression, is now enrolling. Safety updates will be given next year early, and then the next clinical update will be the 3-year time point, again with matched historical control data. Thank you.
Thank you very much, Ed, for this nice update despite your life-threatening injury. So, are there any questions?
Everyone else needs the mic. I can hear you, but they won't.
Kia ora, thanks for that. I'm Cindy Towns. Not sure if you're aware, but we've recently established an ethics advisory board, and we've been discussing placebo controls at the Advanced Therapies Working Group, certainly for those of us who didn't unfortunately get stuck in Madrid for 24 hours, long story, won't go there. So I'm interested in the justification for denying you a placebo. We don't tend to call it sham anymore, but and I'm wondering whether that was based on the science, whether they thought that an open label trial would give you the same results, or whether they elaborated and had some sort of black and white ethical opinion on that. I'd love to hear about it.
So I'm happy to connect you directly with the sponsor for a more detailed discussion about that. But in effect, the FDA insisted on the placebo surgery arm. The EMA insisted on not having the placebo arm. And so there was an enforced divergence of the approaches. Of course, the placebo surgical data from the US 12-month patients can be used to compare against the European data, but we don't have contemporaneous, geographically concordant participants. As I know you know, because I had the update from that working group on Tuesday, it's a very controversial area, and with, I think, extremely persuasive and emotive issues on both sides. And I hope it's one where we can at least obtain a kind of agreement across the Atlantic about how to think about these issues, because it's a real challenge. But even if you do have the placebo surgery, the time duration over which it can be used is pretty limited, and there is also a lot of debate about what that surgery should consist of. So a skin incision, a drill hole into the outer table of the skull, something deeper. You know, I mean, in theory, the ideal placebo control would be a scrambled siRNA inside a virus that you inject into the striatum. But of course, that's a hugely challenging prospect. In other programs that have recently been announced in the US, the FDA has insisted on unilateral treatment at baseline, with a period of observation to compare left and right, and then, if safety permits, you go back into theatre and have another neurosurgical procedure for the other side. I have no skin in the placebo game except to say data is good, and these are extremely challenging questions.
Okay, thanks, Ed.
Merci.
Thank you so much. Certainly a fascinating topic, and maybe we should continue it after the clinical trial session. Myself had a question for Ed Wild. Now I would like to transition to a different program. So Dr. Peter McClogan is Global Development Leader of Roche. It's a pleasure to have you.
Thanks so much, and thanks for the opportunity to share some of this data today and to give an update about where we are with our current trial. So my name is Peter McClogan. I'm Global Development Leader at Roche. And I want to share some exciting new fluid biomarker data we have from our previous study, Generation HD1, and put it in context what it might mean for our current study, Generation HD2. I'm also going to provide an update about where we are with recruitment for our current study and also the outcome of the recent IDMC review. I'll just begin with my disclosures, and then here's an outline of my talk. So first of all, I want to explain what YKL40, total tau, and GFAP in the CSF can tell us. Then I want to share the data from Generation HD1, where we can look at those with the highest doses or the highest exposures in Generation HD1 and the lowest exposures, and look at the biomarker profiles along that. Then I'll give an update about recruitment and IDMC review for Generation HD2. So what can YKL40, total tau, and GFAP in the CSF tell us? We need to first consider the cells in the brain. So we have neurons, we have microglia, we have astrocytes. And if we think first about YKL40, this is a measure of neuroinflammation. So it's secreted by microglia and associated with astrocytosis. Then if we consider total tau, so this is a non-specific marker of neuronal death. It's not dissimilar to neurofilament light or NFL, which I'm sure many of you are familiar with. Then we have p-tau181. This is specific to aggregated tau. Then GFAP is a measure of astrocytic reactivity. But importantly, what do these measures mean in the natural course of HD? So there's a lot of data in the literature from Professor Ed Wild's group at UCL and others that show in HD, YKL40, total tau, and GFAP increase relative to healthy controls. So it's reasonable to assume that an increase may be a bad thing and a decrease in these measures is potentially a good thing. So let's look at the exposure response data from Generation HD1. I'm sure many of you are familiar with Generation HD1, but for those that aren't, this was a phase 3 study. We had individuals that had 120 milligrams every 8 weeks, 120 milligrams every 16 weeks, and placebo. What we do in an exposure response analysis is we pull these individuals together. We can measure the level of drug or tominersen in the CSF. So we can then split people into 4 groups, those with the highest exposures, those with the lowest exposures or level of the drug. And we can compare this to placebo. So if we start off by looking at CSF NFL here in purple and CSF mutant Huntington in pink, you can see that at the highest exposure, NFL goes up, it peaks at week 21 and comes back down again. And you have quite dramatic mutant Huntington lowering of the realm of about 50%. Now, if you move down quartiles to the lowest exposure and you compare this to placebo beside it, you can see that you can avoid that NFL increase, but you still have mutant Huntington lowering in the realm of about 25 to 30%. And importantly, this lowest exposure quartile is the quartile where we expect the new doses in Generation HD2 to be. So this potentially gives us an insight into what we can anticipate in the new study. Now let's add in YKL40. You can see this here in pink. At the highest exposure, you can see YKL40 goes up and comes back down again. At the lowest exposure, you avoid the increase. So similar to NFL. Then let's add in total tau and p-tau181. Again, at the highest exposure, you can see it goes up at week 21, comes back down again. But at the lowest exposure, you avoid that increase, and you actually see it go below baseline. And I'm going to zoom in on this in a couple of slides. next. Now let's look at GFAP. GFAP, again, at the highest exposure it goes up, but importantly at the lowest exposure, where we're targeting the doses in the new study, you can see that p-tau181 and total tau actually go down. So let's zoom into this low exposure quartile. Here we're looking at placebo in black, and in the blue we're looking at the low exposure quartile. Let's start with CSF mutant Huntington. You can see that we're lowering by about 25 to 30% at trough. CSF NFL, you can actually see it trends below placebo. YKL40, it trends below placebo. Total tau and p-tau181, again, it goes below placebo. With GFAP, it increases, but then it comes back in line with placebo. Now, this suggests that at this exposure, again, the exposures we're targeting in the new study, we are seeing potentially favorable biomarker profiles. So, of course, this is an exploratory analysis, but it really provides confidence and some excitement as to we think we're in the right place with the new doses. So let me just summarize. CSF YKL40, total tau, and GFAP can tell us about neuroinflammation, neuronal death, and astrocytic reactivity. At the highest exposure, you can see these go up, but at the lowest exposures, we avoid the increases, and with some of the biomarkers, you see them go down below placebo. So this is really exciting, really promising for moving forward. Now let me switch gears. I want to first give an update about recruitment, and then we are with IDMC. But for those that may not be familiar, let me just give an overview of Generation HD2. So this is a phase 2 study evaluating safety biomarkers and looking for trends of efficacy in 2 doses of tominersen in individuals who are prodromal or early manifest HD. We're looking at 100 milligrams every 16 weeks, 60 milligrams every 16 weeks, and placebo. Individuals complete 16 months and then they follow a common close. That means that they continue on with blinded treatment until the last participant has had their last visit. And the IDMC look at the data every 4 to 6 months. So what is the IDMC? This is an independent group, independent from Roche, including an expert statistician, a brain imaging scientist, and Huntington's disease experts that look at safety and clinical outcomes and assess the balance of benefit and risk. And they're looking at safety, clinical outcomes, and brain imaging data every 4 to 6 months. Importantly, the IDMC can recommend either to continue, modify, or stop the study. And so I am delighted to say at the last IDMC review, they recommended for us to continue as planned. They looked at data from 219 participants, up to 5 doses of study treatment, at an average time on treatment of 166 days. And as I mentioned, they looked at safety, clinical, MRI, and plasma NFL data. So this is very reassuring. And I also want to say that we're more than 80% recruited, which is fantastic. And we plan to complete recruitment by the end of the year. And with that, I'd like to say a massive thank you to all our collaborators, but most importantly, patients and their families, because without you, we couldn't be where we are today. So thank you.
We are open for questions.
The informed consent papers had to be amended with additional information about the risk of tominersen. What is the philosophy not distributing that information to other European countries?
Thanks, Dr. Langer, for the question. So we take feedback very seriously, and we want to ensure that everyone is adequately informed. All of our trial documents go through ethics in every country, and we get feedback from those ethics boards, and we take that seriously, and we can change things based on that. So if there's any concerns about local countries, I'm more than happy to talk to people afterwards. But as I say, everything goes through health authorities and ethics reviews boards, and if there's any concerns, then we can adapt stuff based on that. I hope that answers the question. But happy to catch you up after as well.
Thanks, Peter. It's more of a comment, really, than a question, just to say that I think what Roche has been doing really vindicates, the philosophy of looking really carefully at clinical trial data, even if it's negative. So I'd just like to congratulate you for doing that so carefully. I think it's been really valuable.
Thank you.
So, if there are no more questions, thanks again, Peter. Okay, so then we move on to the next speaker. It's a pleasure to welcome Dr. Jane Atkins she's a Senior Vice President at Wave Life Sciences. So, welcome.
Thank you. Good morning. I'd like to thank the organizers at EHDN for giving me the opportunity to share with you the results of our SELECT-HD study today. Okay. Forward. I'm going to be making forward-looking statements, and I'm an employee of Wave Life Sciences. So in the next maybe 7 to 9 minutes, I'm hoping to leave you with 4 key takeaways from this talk. The first is that WV003 selectively and potently reduced mutant Huntington levels to a level greater than 30% in the CSF. And it did this in an allele-selective manner. It preserved wild-type Huntington. These 2 points, along with the encouraging safety and tolerability profile of 30 mg, warrants further clinical development of WV003. And lastly, what was really exciting for us is that we saw for the first time an association between mutant Huntington lowering and the slowing of caudate atrophy. So let me start by giving you a little overview of WV003. So 003 is a first-in-class allele-selective oligonucleotide that targets mutant Huntington. It does this by targeting a single nucleotide polymorphism, or a SNP, that's associated with the mutant Huntington allele. And we call this SNP SNP3. 003 works at the level of the transcript to reduce the amount of mutant Huntington protein that is produced. And because it's allele selective, it spares the wild-type allele, allowing expression of wild-type Huntington, which is a very important protein that we all know is so important for neuronal health and function in the CNS. So turning now to the design of SELECT-HD. SELECT-HD was an adaptive phase 1b/2a study that was designed to rapidly assess WV003. It consisted of 2 parts, a single ascending dose phase that looked at single doses of 30, 60, and 90 mg, and a multi-dose phase that looked at 30 mg given 3 times 8 weeks apart. The objective of the study was safety, but we did look at exploratory biomarkers, including mutant Huntington, wild-type Huntington, and NFL, and we also looked at clinical assessments. Because of the time that I have allotted for this talk, I'm going to be focusing the majority of the rest of the talk on the results of the multi-dose phase. Baseline characteristics were well-balanced across the study. In terms of safety, 30 mg was generally safe and well-tolerated throughout the study. All of the AEs at the 30 mg were mild to moderate in intensity. We had no SAEs in the multi-dose phase. There was a slight imbalance in the treatment-related adverse events in the multi-dose that were all mild to moderate. The mild events included hyperreflexia, and the moderate events included laboratory CSF abnormalities, including CSF protein elevations. Turning now to the biomarker data, WV003, multiple doses of it, selectively knocked down mutant Huntington to a maximum mean of 46% at day 169. This reduction was durable, as at day 197, it was still reduced by 44%. When we looked at the wild-type protein, we were really excited to see that the levels of wild-type did not go below baseline throughout the duration of the study. So these 2 graphs confirm the intended mechanism of action of WV003. It works. So taking these 2 with further PK/PD modeling that we've done also we're excited to say, supported moving forward to the more convenient dosing regimen of quarterly dosing. We did look at NFL. This is a graph showing the individual patient profiles of NFL in the multi-dose phase. The gray box outlines the 5th and 95th percentiles of the placebo-treated groups. So a couple of things about this graph. The majority of the patients were within the placebo range. There were a few that went above placebo. By and large, those that went above placebo tended to have higher exposures of 003. We anticipate that going to the more convenient 12-week dosing regimen will mitigate these increases as the overall exposures will go down a little bit. Importantly also, when we looked, we saw no correlation of NFL with the number or the severity of AEs. We also saw no correlation with caudate atrophy or ventricular volume. In addition, when we looked at plasma NFL, we saw no significant difference between placebo and treated. Very encouraging. SELECT-HD was not powered to look at MRI or clinical assessments. However, we did see some results that were very informative and exciting. So looking at normalized caudate volume, when we looked at the change of caudate at the end of the study, we were really excited to see that those treated with 30 mg of 003 had slightly less caudate atrophy at 4.68% than those treated with than those in the placebo group, 5.1%. In addition, we saw a statistically significant correlation between the amount of mutant Huntington lowering and caudate atrophy. This is the first time this has been shown in the clinic, and we were super, super excited about this. We also saw a correlation between increasing 003 exposure and the slowing of caudate atrophy. Again, we looked at clinical assessments. The study was not powered to look at these, and so we weren't surprised that we did not see a statistically significant difference on any of these: CUHDRS, SDMT, TFC, and TMS. We were, however, pleasantly surprised to see a point estimate improvement in TMS that favored 003. So while this is not statistically significant, this point estimate difference of about a little over 4 points is actually greater than the MCID of around 2 to 3.5 points. So it's really encouraging, and we're really excited to see this. So taking you back to the 4 points that I started out with at the beginning of the talk, 003 selectively and durably knocks down mutant Huntington, and it does this in an allele-selective manner that supports a dosing regimen of every 12 weeks. It's safe and tolerable, and we also, for the first time, showed a demonstration that mutant Huntington lowering correlates with the slowing of caudate atrophy. So we are very excited about these results. We are now moving forward and actively engaging with the FDA, and we hope to have information on the path forward for the next stage of development by the end of the year. Thank you.
Thank you for sharing these exciting results. Are there any questions from the audience? I think
there's
a question. Yeah.
Hi, Jane. Thanks. Nice, very nice presentation.
Where are you?
Right here.
I don't know where right here is.
Okay, there.
Oh, there you go.
What are WAVE's estimates for what percentage of HD patients will have SNP3 attached to their mutant allele, roughly?
Right now, our assessment is about 40%.
Okay.
Jane, over here, Ralph.
Okay.
Congratulations to the data. I think it looks great. Yeah. One question was this backbone. Do you have anything in development for SNP1 and 2? Because it would be nice to revisit those.
Oh, yes, absolutely. So if we get feedback from the FDA that enables us with a favorable path forward, we absolutely would go and look at other SNPs and go back and start to renew this improved chemistry on those SNPs so that we can, of course, address the greater HD patient population.
So there's also a question.
Thank you. Here on the front.
There we go. Sorry.
Yeah. Thanks for the presentation. Your SNP is targeting, I think, exon 59. What is your thinking of the effect that you can envision on exon 1 of Huntington?
Exon 1. So theoretically, we are, we are knocking down the transcript. We're completely allowing it so that the transcript is gone. So theoretically, we would be reducing the amount of exon 1A that is produced.
Thank you.
Okay, thank you. And there's one question there.
That's good.
Okay, sorry.
Can I go first? I have the microphone already.
Over there.
Yeah, so you go ahead and then...
Okay. So I'm sitting over here in the, yeah. I have a question about targeted reduction, because it's allele specific and you have now a reduction of 50%. What is it you want? So is the 50% reduction what you want from the mutant Huntington? And are there thoughts on loss of heterozygosity? Of what would it mean?
Okay, I missed that second part of the question.
Yeah, please.
I missed the second part of the question, but so we expect, we were aiming for greater than 30%. That's the amount that we felt was needed for clinical benefit. With the Q12 dosing regimen, we anticipate getting 35%. Right, and I forgot, I'm sorry about the heterozygosity, the second half of your question.
Yeah, so you don't want to pull down mutant Huntington for 100%. Because you still have the wild type.
Well, I don't think we don't need to, right? We don't need to.
Okay.
Okay, so let's move on to the last question.
Hi there, Fiona Kinsella from UCL. Can you comment on whether the white blood cells in the CSF or the proteins were elevated at all?
Yeah, so we saw no statistically significant elevations in white blood cell count. And the CSF proteins, we saw some elevations, but they're not of concern. They were generally within line with placebo.
Thank you.
Okay, and last but not least, I would like to invite Dr. Emily Bredlau from, she's a Senior Medical Director at PTC Therapeutics. So,
Thank you so much. I'm happy to be here speaking on behalf of PTC Therapeutics today, and I'm excited to share with you an update to the data on the PIVOT-HD trial. So PTC 518 is an HTT-lowering splicing modifier that we're testing for Huntington's disease. PTC 518 brings in an inducible pseudoexon into the HTT pre-mRNA, which leads to a stopping of the translation and mRNA degradation. Importantly, PTC 518 is not allele specific, so it does lower both wild type and mutant Huntington protein. The PIVOT-HD study enrolls participants with HD-ISS stage 2 or mild stage 3 Huntington's disease. They're enrolled into a 5 milligram, 10 milligram, or placebo cohort and receive study drug for 12 months. In the first 3 months, our endpoints include, of course, safety and tolerability, but also blood, MHTT lowering, and CNS exposure. As the year completes, participants are still followed for safety and tolerability, of course, but we're also evaluating CSF, MHTT lowering, blood, HTT lowering, and some clinical scales as well as CNS biomarkers. When participants complete their 12 months of participation, they may, if they choose, roll over into a long-term extension study. This study has no placebo, so participants receive either 5 or 10 milligrams of the study drug, and this extension study goes for at least 2 years. What we'll be discussing today is the first cohort of participants who enrolled onto the study. They were all in the HD-ISS stage 2 cohort, which is why you see here their TFC total functional capacity is 13 for everybody. And you also see that the treatment groups are well balanced. In this participant group, we were able to look both at month 3, as discussed, and at month 12 for their blood MHTT lowering. And you see that we found a durable and dose-dependent impact on mutant Huntington lowering through month 12. The team believes that we reach steady state of HTT protein lowering between 6 and 9 months of study drug exposure. In a small subset of these participants, we were able to measure their CSF MHTT lowering, and as shown in other talks this morning, the mutant Huntington protein level in the CSF can be very, very low and even undetectable in some early stage patients. And so here we're showing only the median of lowering. There just aren't enough participants to share the mean at this point. But we were happy to see that the CSF protein lowering is consistent with that demonstrated in the blood. We were also very happy to see that our NFL levels were steady through the 12 months for all of these participants. One might even imagine some lowering in NFL, but it's really too early to commit to that statement. At the very least, we're very happy not to see any spikes in the NFL in these patients. We were also very excited to see an improvement or a slowing of progression, hint a trend in the total motor score. Again, small cohort, much more to be learned, but this certainly is reassuring. We saw a similar dose-dependent delay in progression in other key milestones or clinical measures, including the cUHDRS and the total functional capacity. And with regards to safety, you see in this cohort of patients, the adverse events were very well balanced between the groups and were reassuring. The most common adverse events were nasopharyngitis, influenza, headaches, and falls, all consistent with the patient population. So we're very happy to say that so far we're seeing a durability of effect, we're seeing reassuring safety, and a dose-dependent benefit may be demonstrable in our clinical measures. Participants are ongoing in the PIVOT-HD and the long-term exposure studies, and I would love to share with you, this is brand new news. We have an INN, an international non-proprietary name for PTC518. It's Votoplam. I'll say it again because it's a new word for all of us. Votoplam. All right, and I'd just like to conclude with a great big thank you to the scientific community, patients, and patient families for all of the support that has gotten us this far, and I'm happy to take questions.
Thank you very much. We have time for questions.
5 minutes.
Are there any questions?
Oh, I've answered all of the questions.
Okay, so there is a question at the back.
Hi, thank you for this update. This is great. It's Danielle from Vanderbilt University. Can you hear me?
I can now.
Oh, sorry. Thank you for the updates and the results. This is great news. This is Danielle from Vanderbilt University. And I was just wondering, are there any future plans to open up the study in North America again?
Yes, thank you. So as many of you may know, our phase 2 study was not open in the United States, but we are very happy to anticipate with, we anticipate opening this study worldwide, including the US. Our conversations so far have been very encouraging.
Any other questions? I think there is one there.
Thank you very much for your presentation. Have you observed any drug-related systemic effects, adverse events?
I'm sorry, I'm having a hard time with the audio.
Have you observed any drug-related systemic adverse effects?
Oh, well, I mean, we've treated 150 participants for almost two or 3 years at this point. So certainly some have been considered drug-related by investigators. I think what you're actually asking me, though, is are there any safety concerns on the study?
Well, yeah, that's correct. Because, I mean, given that this is orally administered, and highly likely there can be any off-target effects of this medication. So whether any off-target effects, adverse effects have been observed?
Okay, thank you for the clarification. So, we're really fortunate that the team was measured in their selection of PTC 518 or Votoplam. Got to get used to that, in their design. So in order to achieve on-target toxicity, we would have to increase the dose substantially because of the design of the drug. So it isn't anticipated for Votoplam, and it isn't seen on the study.
So is there any other questions?
I think we have a hand down here at the very, very, very bottom.
Okay, there is a...
Anything else?
Keep coming.
Yeah. Hello, Jean-Louis Mandel. This is actually maybe more general question to people doing HTT lowering that is not specific for the expanded allele. And I was wondering whether it has been examined what is the clinical status of people in UK Biobank who have, there are a few I think, who have actually loss of function heterozygous mutation in HTT, because this could be informative given the good data in UK Biobank and what happened to these people, and they are 50, 60, 70. I think I don't know if this has been done, or may be interesting to see what is the long-term effect of having just one a dosage of one rather than two HTT.
I think that question was more for you than for me.
Okay, I would be happy to elaborate. So, to my knowledge, that hasn't been done yet, but we know from animal experiments, of course, that Huntington knockout mice or Huntington knockout is lethal.
Yeah, but I'm speaking of heterozygous, because you are not totally destroying HTT synthesis. But what happens when you have 50% of HTT, because you have been lowering both the mutant and the wild type, so it could be equivalent to maybe being heterozygous for loss of function, and what is a long-term effect, not at the homozygous state, but at the heterozygous.
Yeah, I think a very interesting idea. To my knowledge, that hasn't been done. So, yeah.
Yeah, I agree. It's a very interesting question. We're certainly being careful not to over-lower the wild-type Huntington on this program, and we'll be watching. It's a good question.
So any other final questions?
Final question, yes.
Sorry, can I? Sorry. Hey, sorry, Robert Pasivasi from PTC, is from CHDI. Freudian slip. It's a great question you ask, and one that we've looked at. Unfortunately, the data are incredibly limited. There are a very few individuals that have about adults that haven't been extensively phenotyped that seem normal. So that's why most of the people seem to think that in adulthood you can probably get away with 50% of Huntington. That's not in a mutant context, so we don't know, but we very much would like to explore resources like the biobank to be able to look more extensively at that question.
Very good. And we're running out of time.
Okay, Fred Soudou.
Maybe off stage, you can. Okay. We're really running out of time, I'm sorry. I record the enthusiasm for the questions. I think we were able to keep up with time. It's a wealth of development programs, and that's, I think, in Huntington we're so fortunate of having that. I thank all of you that came to share the most recent developments of all the respective programs. I also record that there's some interesting aspects of study design that would merit a lot of discussion in terms of choosing historic controls versus placebo, but we don't have time to discuss that. Likely during the conference we'll do that. And with that we close this session. The next session is new upcoming clinical trials. So don't go away and stay for looking at the future further. Thank you.
Great.